agent. Ayan. If we use mild oxidizing agent, we will only convert the aldehyde functional group. Okay? Because much easier pong ma-convert or ma-oxidize si aldehyde functional group. And then if we use a relatively stronger oxidizing agent, of course, we will also convert the remaining or the alcohol functional group at the end of your monosaccharide structure. Sir, bakit yung ibang hydroxyl group po hindi na-convert? Because it is not readily available in the solution. Okay? They prefer more the intermolecular forces of attraction between each other. Alright? Again, ang product, when we only converted the aldehyde functional group to carboxylic acid, we call this aldonic acids. Now, if we both converted your aldehyde and alcohol, we call this aldaric acid. So strong oxidizing agents that can be used here include your nitric acid. Alright, so for the third and fourth reaction, supplementary na lang to class. And these are actually lab techniques na po kasi to. So for wall degradation, this is used in order to elucidate the structure of your carbohydrate. But of course, medyo tedious siya sa lab because this is a series of reaction. Alright? So of course, degradation meaning we will decrease the carbon chain in every reaction by 1. And that carbon chain is the one nearest to your aldehyde functional group po. Alright? So for our fourth reaction, kung may degradation, we also have synthesis. Okay, for synthesis, similar with degradation, ang point of attachment ng new carbon chain is the one nearest to aldehyde functional group. Okay, so here we use mainly the reagent sodium cyanide. You will see or you already seen this in your reactions of your carbonyl compounds in organic chem, right? So class again, we will form two different isomeric forms of products because in carbohydrates, hindi talaga sila stereo-specific. Alright, so now we are actually done with our discussion on monosaccharides. Class, before we transition in our discussion of other classifications, disaccharides and polysaccharides, let's first discuss the bond that connects them, and that is your glycosidic bond. Alright, so si glycosidic bond class, covalent bond po yan. So meaning strong bond between your two monosaccharide units. And the reaction is classified as dehydration reaction. Meaning, after the reaction, we liberate a water molecule. So this is a typical example of the formation of glycosidic linkage or glycosidic bond. So here, you can see that for our first monosaccharide, the hydroxyl group directly attached to the the anomeric carbon will be liberated as well as the hydrogen atom of one of the hydroxyl groups of the second monosaccharide to form the alpha-1,4 disaccharide. Sir, important ang naming for your glycosidic bond formation? Yes. That's why, di ba kanina, I emphasize that you need to familiarize alpha and beta and their configuration. Check here if this is really alpha-1,4 disaccharide. So here, is the C1 position. And we know class that always anomeric po ang C1 position. So meaning, lagi siyang may prefix na alpha or beta. Class again, nasa C1 position po ang anomeric carbon. Okay? So that's why nandyan ang prefix natin na alpha or beta. And then the second sugar, it only contributes a hydroxyl group, particularly a hydrogen atom, typically connecting at C4 or C6. Alright? So this one is alpha because this one is pababa. Alpha, baba. Again, ang i-check nyo lang ay yung C1 carbon. Si C4, this is not anomeric. That's why it's not okay. We will not use the alpha and beta anomeric prefixes. Alright, so I hope that's clear. Now, you can see here that upon formation, yung first monosaccharide unit natin, wala na siyang available anomeric carbon o hindi na free yung anomeric carbon. Hence, unreactive na siya. For the second monosaccharide unit, you can see that hindi pa reacted ang anomeric carbon niya, meaning it still has its hydroxyl group. So, sir, capable pa po ba siya na maging acyclic or mag-undergo ng mutual rotation? Yes. Then we call this specific monosaccharide unit as reducing because it can still react with numerous reagents. And dito class papasok ang discussion natin on reducing and non-reducing sugar. Formally defined as reducing agent due to its free aldehyde or ketone functional group in its structure. And that is your reducing sugar. Sir, meron bang example ng non-reducing sugar wherein both the anomeric carbon participate in forming the glycosidic bond? Yes po. And this is very common in nature. Familiar kayo class with the structure. This is actually your sucrose or your table sugar. As you can see, yung both anomeric carbons, they participate in forming your glycosidic bond. So class again, ang C1 position is always anomeric in your aldoses. For your ketoses, that is on your C2 position. O recall ang monosaccharide unit kasi ng sucrose is glucose and fructose. Sir, can we identify their glycosidic linkage nature? So let's check. For the first monosaccharide unit, si C1 ang nagpa-participate. Check, is this alpha baba or beta as? Class, alpha baba yan. That's why it's alpha 1. And then here for our C2 position, beta as. Because again, above the plane po si oxygen for this particular glycosidic linkage. That's why for your sucrose, its glycosidic linkage is labeled alpha 1 beta 2. Okay, class, in order to practice you in glycosidic bond formation naming of its connection, so we prepared a sample problem. So I think nasa manual nyo na rin siya. So for this discussion, we will answer it here. So again, identify the type of glycosidic bond. Sir, can you give us a tip? First thing to do here is to identify the carbons that participate in the glycosidic bond formation. So for our first monosaccharide unit, that is your C1 position. Sir, paano mo nalaman na C1? Ang technique ko dyan, guys, the C1 position is always directly bonded to the oxygen inside the ring. Okay, so meaning sir, yung second monosaccharide unit, C1 din po ba yan? Yes. Alright? So that's why C1 ang nagpa-participate sa bond formation natin. And again guys, as we have discussed earlier, for your aldoses or aldohexoses in particular, C1 lagi ang position ng anomeric carbon. That's why for both monosaccharide units, we will include prefixes. So sir, meaning we will just determine for the two of these labels, the alpha and beta configuration. Yes. Okay, so for first monosaccharide unit, is this alpha or beta? Clearly, class, this is an alpha baba because again, below the plane of your aldose. And for the second one, it is also alpha baba. So both of them is alpha. That's why we label the glycosidic bond alpha 1, alpha 1. Okay, so this is actually your three halos sugar, also known as your zombie sugar. And because of both anomeric carbon participates in forming your glycosidic bond, this is non-reducing, meaning wala na siyang free anomeric carbon that can undergo mutarotation or reaction. Okay, so number 2. Ayan class. Again, same tip. Label the two carbons participating in your glycosidic bond. For the first monosaccharide unit, sir, alam ko C1 yan because again, directly attached siya sa oxygen inside the ring. Yes po, very good. So for the second monosaccharide unit, naku sir, hindi yan C1 position because this is the C1 position. C1, C2, and then C3. So sir, hindi na namin need lagyan niya ng prefix na alpha or beta, no? Yes, that's why nasa C3 position siya. O class, now, we formally name the glycosidic bond as alpha or beta. So check the point of connection. You can see here, it's below the plane of the ring. That's why alpha 1, 3 ang glycosidic bond for this specific sugar. And we call this nitrose. So class, si nitrose, this is found in small amounts in dextran polysaccharides. Also reducing pala yan because again, free ang anomeric carbon niya. So reactive pa po siya class. Alright, so that's it for our two sample problems. So class, hinga muna kayo. We are transitioning to next topic. So because we are already done with the simple carbohydrates. So class, remember ha, si simple carbohydrates, those are just your monosaccharides. Meaning, hindi pa natin siya kayang i-hydrolyze because that's the basic building block of your carbohydrates. For our next discussion, we will tackle complex carbohydrates. So this can be your disaccharides, oligo, and polysaccharides because it is linked together by glycosidic bonds. So class, recall lang ha, ang mga nagamit na natin na visualization structure for our discussion. So we have your Fisher projection for your aldose and ketose. We also have your, in your organic chem, what do you call the structure? Hayward projection. And then lastly, in order to easily visualize polysaccharides or complex carbohydrates, we use, ayan, the chair conformation. Okay, so now let's proceed with our brief discussion on disaccharides. So for disaccharides, polysaccharides, or in general, the complex carbohydrates, this is just really going to be a trivia discussion. Because again, class, in your board exam, trivia heavy po ang biochem. So meaning, if you don't know the answer, you cannot answer it correctly. Okay? It's either a hit or a miss. That's why we need to cover a comprehensive amount of topic here. Baka kasi yung iba sa inyo, similar with our on-site classes, nagtataka sila, bakit different ang discussion here at QRC Biochem compared to their undergrad? Because again, class, we tailor fit already our discussion to obey the TOS from PRC. So we omitted unnecessary information para efficient ang discussion. Alright, so going back with our discussion, this is your disaccharides. Alright, so oligosaccharides are composed of 2 to 10 monosaccharides. So, sir, confusing. Akala ko disaccharides. Actually, class, there is an overlap kasi with the definition of these two. Si disaccharides, limited lang po siya sa sugars that is composed or that are composed of 2 monosaccharide units. Alright? So, si oligosaccharides is the groupings that is composed of 2 to 10 monosaccharides. So, meaning, sir, umbrella term si oligosaccharide? Yes. And both of them, they are joined by a glycosidic bond. O, class, quick quiz muna tayo. So here we listed the 3 relevant disaccharides and their common names. Now, we will identify their monosaccharide units. First one, table sugar. Na-discuss ko na to earlier. That is your sucrose. Okay? And we know that sucrose from our discussion on reducing versus non-reducing, its monosaccharide units are glucose and fructose. Okay? Class, for the 2 remaining disaccharides, please try answering their monosaccharide units. Alright? So for milk sugar, this is also called your lactose. So ito class, madali nang ma-determine ang monosaccharide units. From the name itself, lactose, one of its component is galactose plus glucose. Alright? And then lastly, we have your malt sugar. From the name itself, maltose. And this is actually just 2 monosaccharide units of glucose. And I think given na po ang structure ng 3 disaccharides in your manual. Or remember their glycosidic bond formation or name ha. Again, for sucrose, alpha 1, beta 2 for maltose, alpha-1 for lactose, beta-1,4. Sir, may importance ba yung alpha and beta anomeric forms? Yes, because the functions of your disaccharides and polysaccharides are dependent on the nature of their glycosidic bond. Okay, so moving forward, ayan. So again guys, similar content lang yan from the previous slides. But here, we have an additional oligosaccharides. Sir, do we need to study that? Yes, but no need to memorize, okay? This just shows that we really have a long list of disaccharides and oligosaccharides, okay? So we will give you a copy of this slide. But for now, let's move forward with our discussion on polysaccharides. So again, class, all you need to remember here in the disaccharide discussion are the three relevant disaccharide examples. Okay? So now, ayan, polysaccharides na class. Konti na lang and we will finish our discussion on carbohydrates. So polysaccharides, they are the grouping of your carbohydrates in which they contain three or more monosaccharides linked by glycosidic bond. All right. So of course, similar with your monosaccharides, they can also be classified into two broad groups, si homo and si hetero. Sir, anong difference? Class, kapag homo, meaning yung monosaccharide niya is just one type. Okay, so for instance, here we have your amylose of starch and cellulose. Both of them, their monomeric unit is only made up of glucose. Their only difference is in their glycosidic bond nature. O sir, paano naman pag hetero? Meaning po ba niyan, it consists of two or more types of monomeric units? Very good. Okay, so for instance, we have your pectins and gums. Okay, for the succeeding slides class, I will showcase to you the three relevant example of homo polysaccharides. Okay, so walang kukurap. First one, we have your starch. All right, the starch class, always remember it has two major components, and it's their only difference is in the extent of their branching. These are your amylose and amylopectin. Class, remember, si amylose ay only 20% ng starch and si amylopectin is 80%. That's why very good energy storage si starch in your plant organisms. Okay? So again, in terms of the glycosidic bonds, si amylose po, it only has alpha-1,4 glycosidic bonds. And the branching of your amylopectin, sorry class. So amylopectin arises from your alpha-1,6 glycosidic bonds. Okay, again, si amylopectin, similar lang po siya kay amylose, may additional alpha-1,6 lang for the branching. So I hope that's clear. Klaro ba class? Ayan, so very easy for starch. Now, we have also your cellulose. O si cellulose class, nasa cell wall yan ng almost all plants. And of course, its main structure is its main role is for structural support. And it also gives rigidity to wood and plant stems. O sir, ano po bang structure ng cellulose? Ayan siya class. O di ba class, kanina I emphasize the nature of your glycosidic bond has an effect in your polysaccharides. Ito class, beta-1,4 ang glycosidic bond ng cellulose. That's why it can efficiently pack itself. And that's why ang role ni cellulose is mainly for structural support. Okay? So, sir, water soluble si cellulose, no? Because of the numerous hydroxyl groups? No. Okay? Ang hydroxyl groups po ni cellulose is not or are not available because of its tightly packed structure, making it water insoluble. Okay, class, again, beware, not all polysaccharides are water soluble. First reason, maybe hindi available ang hydroxyl groups for hydrogen bonding. And the second reason is that yung hydroxyl groups are on a strained position. Okay? Not readily available. Alright, so class, ayan, meron tayong did you know segment. Here in Biochem, all did you know segments are board exam concepts. That's why I will highlight it in our did you know segments. For this segment, we will focus on cellulose. Not all carbs are created equal. Why? Because humans can digest starch, but we cannot digest cellulose. Okay? Because we lack the enzyme that degrades cellulose. However, class, merong organisms, particularly mammals, that we call ruminants that are capable of digesting cellulose because first, they have got microbes that can release cellulases, which is the enzyme capable of degrading cellulose. And lastly, they also have rumen, an additional structure in their digestive system. All right, so that's it for our Did You Know segment. And lastly, we have the counterpart of your starch. Sir, paanong counterpart? Si starch po kasi, this is the energy storage of your plant organisms. While si glycogen, as we all know from your biochem undergrad, this is our main energy storage. Sir, meaning this is the one prioritized by the body upon need of energy? Yes. Okay, so this is the structure of your glycogen compared to your amylopectin. Class, observe na much more extensive ang branching with glycogen. That's why more efficient in terms of storing energy in the what do you call this, in the form of glucose units, C glycogen. Okay, again, to formally define this glycogen, this is your branched glucose polymer that is used by animals as an energy reserve. All right, so again, si glycogen, wala po siya sa plant organisms. For us animals or mammals, highly concentrated siya sa liver, but it is also localized in your skeletal muscles. Class, remember the three polysaccharides. Do you have starch, cellulose, glycogen? Sir, tama no? All of them are homopolysaccharides? Yes po. All right, so now we proceed with our last topic for carbohydrates, chemical characterization. Okay, so first, ayan, these are some examples of chemical characterization. So first one, this is your Molish test. Very characteristic ng Molish test, ang purple interface. Next one, we have your Seliwanov's test. Okay, the red positive confirmatory color ng Seliwanov's test. The third one, this is actually your Benedict's test for reducing sugars. And this one is your osazone test or osazone formation, which is very useful in order to complete structure elucidation of carbohydrates or confirmatory of carbohydrates. So here, we have 5 known chemical characterization lab techniques. First one again, Molish test. Positive sign is purple interface. Class here, I'm not going to require you to memorize, okay, to memorize the reagents as long as you are familiar with the positive signs of reaction. All right, going back, we have Biles test. Positive sign, blue solution for pentoses. So this test distinguishes your pentoses from hexoses. Okay? So for Seliwanov's test, ayan, as I have said earlier, red solution po ang positive sign for ketoses. So sir, compared with Biles test, si Seliwanov's test po ba is between ketoses and aldoses? Yes. Kapag red, meaning really meron tayong ketose sa solution. In the case na meron tayong aldoses, of course, retain lang yung original color ng solution. All right. Number 4, we have your Benedict's test for reducing sugar. Class, I will remind you here ha, red precipitate po. And a pale red solution because of that red precipitate. All right, and then lastly, we have your osazone formation. So for all monosaccharides, the characteristic positive sign is yellow crystal. They only differ with their time of formation. Alam nyo guys, very important ang osazone formation in order to determine the specific type of monosaccharide in the solution. That's why it completes the chem characterization of your carbohydrates. Class ha, I will still remind you here na baka mag-appear si Felling's test. So that's the older version of your Benedict's test for reducing sugar. All right, so now, here are my final Lars words for carbohydrates. Class, recap natin ang discussion on carbs. So again, we have here the summary of the structural properties of carbs. Monomer, that's your monosaccharides. Directionality, so this will be discussed more on our carbs metabolism from non-reducing to reducing end. Okay, so naturally occurring forms, again, all naturally occurring sugars, deconfiguration. Energy function, yes. Okay, we metabolize this for energy use. Digestion mechanism. So for enzymes, we use glycosidases and amylases at your mouth and small intestine as sites of digestion. And solubility, again class, this vary per monosaccharides. Pwedeng water-soluble or less water-soluble for your polysaccharides such as cellulose. For classifications, we discussed first monosaccharides. Class, important that you know, we have 2 subclassifications, length of carbon chain and functional group. For pentoses, ayan, meron tayong mnemonics class. Ribs are extra lean, and for hexoses, all altruists gladly make gum in galantangs. And remember, I shared a tip in order to draw the structure of these monosaccharides efficiently. For disaccharides, we again have 3 relevant examples: sucrose, maltose, and lactose. And lastly, we have your polysaccharides. Si homo, meaning similar type of monomeric units, and si hetero, 2 or more types of monomeric units. All right, and with that, ayan, that pretty much sums up our discussion on carbohydrates. Class, please prepare your PT booklets and answer your practice test 1. Remember class please practice closed manual PT answering para ma-simulate nyo na ang mismong board exam. So I will discuss separately the answers to your PTs in another video. Okay, class. Now we will proceed immediately with our discussion of the second biomolecule. Done na tayo sa carbohydrates. Now, we will proceed with lipids. So for this discussion, again, similar structure po. We will first discuss the classification. So we have hydrolyzable and non-hydrolyzable lipids. And lastly here, ang special discussion natin is the supramolecular assemblies of your lipids. So lipid assemblies and structures. And of course, take note, when I say hydrolyzable, it is synonymous with saponifiable. Okay? So it can be cleaved through hydrolysis. So of course, guys, sa carbohydrates, we have seen that they have really unique structure, di ba? In terms of their classification. Here, kay lipids, ang unique sa kanila is their property. Because again, they cannot mix with water. Di ba? We know that they are amphipath molecule. Okay, so before we formally start our discussion, ayan, minigame muna ulit tayo, our quick quest. Similar with our earlier quick quest, so same instruction lang siya. Identify the common theme among the 4 pictures. Sir, ano yung first picture? So this is actually a margarine. All right. Second one is your bilayer macro molecule. Third one, ayan, very familiar. I think this is an olive oil, super healthy. And then last one, this is a drug. Okay, particularly aspirin. So of course, class, the common theme is actually our topic for Crash Course 2. That is your lipids. All right? Last quick quest for our CC2. Ayan, very easy item to class. Okay? It's very bad for our health when took in excessive amounts. And that is your cholesterol. So class, remember ha, si cholesterol is also a type of lipid which we will discuss later on in this Crash Course. Okay, so now here is the overview of lipids. As I have said earlier, class, it is an amphipathic molecule. Okay, so it's capable of storing energy. That's why in our discussion in lipid metabolism, we are also going to touch on ATP accounting. Okay, ang kanyang structure naman, it has a very hydrophobic CC and CH single bonds. Thus, because of this structure, they are soluble in organic solvents and insoluble in water. So, hydrophobic po generally ang lipid. So, here is the overview of its characteristic. Class, among the four biomolecules, I would like to emphasize again that lipids are the only non-polymeric of all the four biomolecules. Okay, so not all of them are also chiral. Some lipids are chiral, those that are already a derivative of them. Okay, sir, need ba naming i-memorize ang sources? No po. It's just here to enrich our discussion. So again, for our sources, we have oil, nuts, seeds, fatty fish, very rich in omega-3 and omega-6 fatty acids, essential fatty acids. All right, so for classification, okay, class, I already introduced this earlier, hydrolyzable and non-hydrolyzable. So their structure are very different from each other. Yung isa may common theme, yung isa is very varied in terms of their structure. Oh sir, di ba si carbohydrates, glycosidic bond ang unique bond sa structure niya? Yes po. Here in lipids, our bond type, our unique bond type is ester bond. Okay class? But of course, reminder, not all lipids have ester bond. And lastly, key function niya po is for structural pa din, cell membrane and structural support. Okay, so let's just recall what does amphipathic or amphiphile mean. Ayan. So this is from your organic chem discussion. So when we say amphipath, it has a hydrophilic and hydrophobic region or polar and non-polar parts respectively. O sir, ano po yung hydrophilic region ng ating lipids? So in general class, sa lipids, we have a polar group consisting of phosphate and glycerol. Alright? And then of course, si hydrophobic part po or region just consists of your FA chains or fatty acid chains. So later on, you will see kasi that fatty acids are just chains of hydrocarbons. This is also the reason bakit hindi polymeric si lipids. Alright, so again, remember class, our term, amphipath or amphiphile. Now, we will proceed with the discussion on classification, starting with hydrolyzable lipids. So when we say hydrolyzable, kaya siyang i-degrade or i-cleave into smaller molecules via hydrolysis. Okay? So ito class yung classification ng lipids that have a common functional group. So lahat po sila may ester unit. Alright, so for our discussion, we are just going to highlight the three relevant hydrolyzable lipids. We have waxes, particularly biological waxes, and then we also have tags. So that's your triacyl glycerols. And lastly, your phospholipids are also known as phosphoacyl glycerols. O sir, nasan po yung ester units? O, di ba? Ang ester units nyo are carbonyl attached with single bonded oxygen. So this is the ester functional group, this is the ester functional group, and this is the ester functional group, as well as here in your phospholipids. Okay? So we will dissect the details of the structure of each hydrolyzable lipids on the next slides. All right, so now let's first discuss biological waxes. O class, from its structure earlier, you can see that waxes are actually the simplest hydrolyzable lipids. So, nag-condense lang tayo ng a part of a fatty acid molecule and a part of high molecular weight alcohol. Alright, so sa industry, particularly sa cosmetics, sa food, it has a very crucial application. Familiar with cosmetics, di ba? For hair waxing. Sa food industry, it gives shine to our gums and candies. Alright? And then lastly, for plant biology, it serves as waterproofing, essential for the functions of our plants and other organisms. Sir, can you give us an example of a biological wax? Ayan. So this is actually your triacontanil palmitate, which is a major component of beeswax. O sir, nasan dyan yung alcohol part? So class, your alcohol part is here. Attached with your alcohol is your fatty acid, long fatty acid. Alright, so that's it for biological wax. Ang hint natin dyan, simplest hydrolyzable lipids. Okay, so class, before I forget again, don't forget to annotate, okay? Use your manuals to highlight or annotate essential information from our discussion. So as you can see, medyo nagpa-pile up na yung information during our discussion. All right, going back, we have triacyl glycerols. So from the name itself, class, it's so easy to deduce the structure of your tags. Triacyl, meaning it has three acyl groups from your fatty acids. So those three acyl groups are here. 1, 2, and then 3, and of course, condensed in a glycerol backbone. O sir, yan yung precursor molecule ng glyceraldehyde, di ba? Yes po. And here, ang ating main takeaway is that tags are the most abundant lipids. Kung si wax is the simplest hydrolyzable lipid, si tags naman is the most abundant lipids. Okay? So we termed it commonly as fats and oils. Sir, anong difference between the two? So it's all in the fatty acids in their structure. Again, class, I would like to reiterate, si tags ay triesters ng glycerol with fatty acids condensed in its structure. So upon hydrolysis, which occurs during lipid degradation, we will obtain two different or unique molecules. First one is your glycerol and the second one is fatty acids. So sir, necessary ba na similar yung three fatty acids in terms of their structure? Hindi po, okay? They can vary. And of course, guys, here in our discussion of tags, very important then that you know the process of saponification. All right? So this is actually a technique in order to produce soaps. Again, your soaps are just made up of salts of fatty acid. So saponification is a process of hydrolysis of your tags in order to produce glycerol and soaps or salts of your fatty acids. Okay? So now, if you are asking, sir, what is the difference between fats and oils? So here, we have a video that will introduce to us the saturated versus unsaturated fatty acids. Because class, again, so saturated and unsaturated are the roots of the difference between fats and oils. All right, so here's the video, class.
Today's video topic is the difference between saturated fats and unsaturated fats. Fat has always had a bit of a bad reputation. People tend to think of it as being unhealthy and something that should be avoided. But the truth is, your body needs fat to function properly. In fact, fat is an essential nutrient. It helps you absorb vitamins, provides energy, regulates body temperature, and cushions and protects your organs. So if fat is so important, why are we told to limit our intake of fatty foods? Well, that is because different types of fat have different effects on your health. In fact, there are two different types of fat, saturated and unsaturated fat, and they both have different effects on your health. What is saturated fat? Saturated fat is the kind of fat that's solid at room temperature. It has all of its carbon atoms bonded to hydrogen atoms. This makes it a saturated fat. It is found in animal products like butter, cheese, and red meat, but also in some plant-based foods like coconut oil and palm oil. Saturated fat has long been linked to heart disease. That's because it raises your bad LDL cholesterol levels and increases your risk of heart disease and stroke. What is unsaturated fat? Unsaturated fat, on the other hand, is liquid at room temperature and is often used in cooking. It has some of its carbon atoms bonded to hydrogen atoms and some bonded to other carbon atoms. This makes it an unsaturated fat. There are two main types of unsaturated fat, monounsaturated and polyunsaturated. Monounsaturated fat is found in foods like olive oil, avocado, and nuts. This type of fat has been shown to lower LDL, bad cholesterol, and raise HDL, good cholesterol, as well as reduce the risk of heart disease. Polyunsaturated fat, on the other hand, is found in foods like salmon, tuna, walnuts, chia seeds, and flax seeds. This type of fat has also been shown to lower LDL, bad cholesterol, and triglycerides, which is a type of blood fat. Polyunsaturated fats also reduce the risk of heart disease and stroke, and also help with blood clotting. So which type of fat is better for you? Generally, unsaturated fat is considered to be the healthier than saturated fat. This is because saturated fat can raise your cholesterol levels, which can increase your risk of heart disease. Unsaturated fat, on the other hand, can actually help you lower your cholesterol levels. It is usually recommended to take about 20 to 35% of your total calories from fat, which is around 45 to 75 grams of fat per day.
Alright, class. So
The
I
American
will
Heart
stop the video. So here, class, ang takeaway natin from the video is that we have two types of fatty acids. So we have saturated and also we have unsaturated. And your unsaturated fatty acids can be further subdivided into two, diba? So as the video explained, we have mono and polyunsaturated. So that's why, class, that is the difference between your fats and oils. So si fats, common component niya or major component is your saturated fatty acids. O, diba? From the video, very unhealthy ang saturated FAs because it will increase your cholesterol levels. Now, on the other hand, si vegetable oils, ang common or major component niya is your unsaturated fatty acid. Okay? In terms of its phase at room temp, solid po ang fat. So for instance, butter and margarine. Si vegetable oils, liquid at room temp. Okay? So for the structure, ito ang structure or visualization ng saturated and unsaturated fatty acid. As you can see, linear ang structure ng saturated fatty acid. That's why relatively longer shelf life po ang animal fats compared to vegetable oils because of the kinks in its structure. So ang reason doon is due to the presence of double bonds. So later on, we will discuss the nature of the double bonds at your unsaturated fatty acids. Plus, check your saturated versus unsaturated fatty acids in the manual. So we summarize the important information that you need to study. But before we discuss that, here is again another comparison of saturated and unsaturated fatty acids. So as you can see here, class, linear ang structure again ng saturated. For example, we have your stearic acid. So this linear chain is because of the lack of double bonds. All right? So as you can see, tight ang packing ng saturated fatty acids. Now, for unsaturated, because of the double bond, we are introducing kicks in your unsaturated fatty acid. Now, ang consequences niyan, of course, it reduces the tightness of the structural packing of unsaturated fatty acid. Okay, class? So for our summary, so again, this is already in your manual. So please take some time to read the content of this table. So for our discussion, I will just highlight the reactivity. So as you can see here, si saturated again, ayan, chemically less reactive because wala siyang double bond. And then of course, because of that, it has longer shelf life. Remember class ha, related po ang reactivity and shelf life. When you are reactive, for instance, like unsaturated fatty acid, you are prone to oxidation. So meaning, mas madali kang ma-degrade and thus you have a shorter shelf life. Sir, saan po galing yung reactivity ng unsaturated fatty acids? So of course, that is because of the presence of double bonds. Okay? So we have common examples for saturated and unsaturated fatty acids. So later on, I will give a mnemonics in order to memorize or familiarize saturated fatty acids. Sir, required ba na i-familiarize? Yes po. So from last year, kwento ng isa sa students namin, nag-appear ang question about what is an example of a C16 saturated fatty acid. O, di ba? Very straightforward, but if you did not review that specific topic, sayang yung 1 point. Okay? So here, again, is a supplementary discussion on the property of your saturated FA in terms of the melting point property. O, ito class, ayan. As you increase the carbon or the number of carbons in its chain, mag-i-increase din po ang melting point. Sir, ano nga po ulit ang reason? Or remember your inorganic chemistry. Again, class, one of the main reasons bakit mag-i-increase ang melting point is because of the increase as well of the IMFAs. Diba? As you increase the surface area of the chain, mag-i-increase din of course ang IMFA. Thus, melting point or the temperature for your melting point will also increase. Now, with our unsaturated fatty acids, we are going to discuss it in terms of the presence of double bonds. Okay? So now here, as you increase the number of double bonds, here, opposite or inverse ang relationship nila. Magde-decrease naman ang melting point. And because of the presence of kinks yan, class. Okay? As you increase kasi the presence of double bond, of course, mas marami yung magiging kinks and it will lead to a very loose packing. Thus, mas madaling mamelt ang unsaturated fatty acids when you increase the presence of double bonds. Alright, so class, here is the mnemonics for your saturated 10 carbon to 20 carbon fatty acids. So ito yung list class. We have cupric, C10, lauric, C12 fatty acid, myristic, C14, palmitic, C16, stearic, C18, and lastly, arachidic, C20. O sir, ano po yung mnemonics? O ito class, ayan, crush lang muna para safe ako. Sir, saan po safe? I don't know class, okay? So mnemonics lang yan, so feel free to use it. But if you have an existing mnemonic, so of course, you can use it as well. So again, for our C10 to C20 saturated fatty acids, crush lang muna para safe ako. Don't worry class, we will summarize the list of mnemonics for biochem. I know medyo mahaba na ang discussion natin. So as we finish the discussion, medyo rarami rin ang mnemonics for biochem. Alright, so now we will proceed with our next topic. Hydrogenation. So from your organic chem, recall, so this is the process of adding hydrogen to your unsaturated fatty acids or to your double bonds. Okay, so hydrogenation, the process will convert double bonds into single bonds. And this is very important in the industry, food industry in particular, to make your fats more solid, stable, and to have a longer shelf life. Okay, class, so usually, ang hydrogenation po is a process that is only applicable to unsaturated fatty acids. All right, so this is an example mechanism of your hydrogenation. O, diba? You have your hydrogen in a metal, solid metal catalyst. And as you can see, ayan, we just converted your double bonds into a single bond to make it a saturated fatty acid. Alright, class. Kasi ang unsaturated fatty acids, that's why marami siyang kinks is because of the configuration of the double bond. So, it's in cis configuration. Class, I would like to emphasize this. Ang naturally occurring fatty acids or those fatty acids synthesized by biological systems, for instance, our body, they are in cis or Z configuration. Recall your OrgChem discussion. So here, when we do hydrogenation, hindi po laging linear ang structure or cis ang fatty acid na na-form. Sometimes, we produce a byproduct known as trans fat. Okay, and this is a very unhealthy fatty acid for the reason that it increases the risk of cardiovascular diseases because again, from the video, it increases cholesterol levels. Okay, so here is the reason bakit ginagawa ang hydrogenation, particularly sa food industry. First one is to modify oil. And of course, second is to lengthen the shelf life. Of course, sa food industry, sorry class, it's very practical to lengthen the shelf life of the food products. So here class, ang products of your hydrogenation. O sir, pwede pala yung partially hydrogenation as seen here in the picture? Yes po. Okay, so partially hydrogenation, we only select double bonds that will be converted to a single bond. Okay, so ito namang bottle at the very right side, ayan, this is the purely or heavily hydrogenated, meaning lahat po ng double bonds na convert into single bond. Ayan, class, kaya please be cautious. Check the nutrition facts kasi again, trans fat and partially hydrogenated fatty acids are not that healthy for us. Okay, so that dapat in moderation lang ang intake natin. Now, done na tayo sa monounsaturated fatty acids. Let's proceed with another type of your unsaturated fatty acids known as PUFAs or polyunsaturated fatty acids. So again, class, lahat po ng double bonds in your unsaturated fatty acids in nature, it's in cis or Z configuration. Okay? So our examples of PUFAs as included in your manual are enumerated here. Sir, do we need to memorize the structure? Hindi po. Ang important here is that you know that omega-3 and omega-6 fatty acids are example of PUFAs. And class, remember, all polyunsaturated fatty acids are essential. So when we say essential, meaning our body cannot synthesize it naturally. That's why we need to take it from our, or they must come from our diet. For instance, oily fatty fish, nuts, and vegetable oils are very rich in omega-3 and omega-6 fatty acids. For those students na curious, paano natin, or paano nag-arrive sa name na omega-3 and omega-6? Okay, meron tayong some organic chem core concept that you need to know. Diba, remember in OrChem, in numbering your carbons in your chain, always prioritized C carboxylic, the carbon in the carboxylic acid functional group. We label it as your alpha carbon, okay? And the next carbon nearby to it is your beta, and then the next one is gamma, until you reach the last carbon of the chain that is labeled as omega. So again, class, yung last carbon sa chain ng fatty acid is called omega. Okay? And with respect to your omega carbon, the first appearance of your double bonds will be the determining factor for the name of your PUFAs. So for instance, kay omega-3, it's omega-3 because the first double bond appeared in the C3 position with respect to the omega carbon. Let's see. 1, 2, and then 3. All right? I hope that's clear, class. So now, we have omega-6. Similar rationalization lang po. Because again, the double bond first appears at the C6 position with respect to the omega carbon. Okay? 1, 2, 3, 4, 5, 6. Sir, ganun pala yun. Yes po. Again, PUFAs, omega-3, and omega-6. So very particular ako here, guys, because nag-a-appear siya every year sa board exam. So I think we already included that specific question in your practice test. All right? So now, we will proceed with our next type of your hydrolyzable lipids. Our last hydrolyzable lipids is are your phospholipids. So we also call them, ayan, phosphoacylglycerols or PAGs, or also known as phosphoglycerides. So recall, class, si triglycerides or TAGs, they are the most abundant lipids. Okay? Second, si PAGs or si phosphoglycerides. Ang main function niya, it is the principal lipid component of most cell membranes. For instance, the plasma membranes of our cells. All right? So for your phospholipids or phosphoacylglycerols, we have two major types. We have your cephalin and we have your lecithin, and both of them can be found in your lipid bilayer. Okay? So here is the structure of your cephalin, ayan, and here is the structure of your lecithin. Again, class, no need to memorize the structure. Just remember that they are the two broad types of PAGs. Ang difference lang naman yan, class, is with their side chain. Okay? So now, for the location, si cephalin kasi, it's more on membrane support. Okay? That's why nasa inner leaflet siya. Si lecithin, it has a role in lipid transport as well as cell signaling. That's why it's found in the outer leaflet of your lipid bilayer. Outer leaflet meaning outside of your cell. Okay? And main function niya rin pala, class, is to maintain the fluidity of your plasma membrane. Alright? So to avoid rigidity. So again, that is the first classification of phospholipids. The second one, ito, class, medyo not known to from our undergrad biochem. So again, si PAGs, it is derived from your glycerol. The second type is derived from sphingosine. So we call it your sphingomyelin. Sir, ano pong structure ng sphingosine? Ito siya, class. So we condense the structure, as you can see, again, similar with your fatty acid. It has a chain of hydrocarbon in its structure. And a common example of your sphingosine-derived molecule is ceramide. Ito, class, very localized sa ating neuron cells. All right? So here is the structure of your second example of sphingomyelins. Ayan. So this is the general structure. So again, this phospholipid is actually a major component as well of your lipid bilayer. And specifically, ayan, si myelin sheath is a type of sphingomyelin that is localized in your nerve cells. All right? So for insulation siya. Very vital component siya ng cell membranes ng nerve cells natin. All right? So these are the three hydrolyzable lipids. Again, we have waxes, we have your triacylglycerols or TAGs, and we have your phospholipids. That's it for your hydrolyzable lipids. So as you can see, class, they all have ester units in their structure. Now, let's move forward to non-hydrolyzable lipids. Class, kapit lang ha. Medyo malapit na tayong matapos on our discussion. For non-hydrolyzable lipids, very varied po ang structure nila because again, they cannot be cleaved through hydrolysis. And for our discussion, we have four biologically important non-hydrolyzable lipids. We have fat-soluble vitamins, terpenes, eicosanoids, and steroids. All right? So sa discussion natin on non-hydrolyzable lipids, I will not require naman to memorize the structure. Okay, ang important here is that you know their unique function and properties. All right, so let's proceed with the first non-hydrolyzable lipid. Ayan. Very known na to, I guess, 'di ba? So fat-soluble vitamins, we already have a mnemonic here eh. ADEC, 'di ba? They are commonly stored in our tissues, fat tissues and liver. So sir, is it okay if we over intake your fat-soluble vitamins? No. Okay, kasi nag-a-accumulate siya inside our body that can lead to toxicity. All right, so again, we have four fat-soluble vitamins ADEC. And for each vitamins, please remember their main function, ha? So for vitamin A, vision. For vitamin D, calcium absorption. For vitamin E, detoxification. It flushes your toxins inside the body. And ito class, it always appear in the board exam. Vitamin K, blood clotting. Okay, very important yan. And later on, we will discuss the role of vitamin K in synthesizing prothrombin for prevention of blood clotting. Okay, further structure, here it is guys. Very bulky po lahat ng fat-soluble vitamins. Okay, no need to memorize the structure. So class, since we are now in the discussion of fat-soluble vitamins or vitamins in general, so I also would like to introduce the B vitamins. But again, I would like to emphasize, the B vitamins po are not fat-soluble vitamins. So we will encounter this frequently kasi sa discussion natin on metabolic pathways as they act as coenzyme in our metabolic reactions. Okay, so we have 8 vitamin B complexes. We have vitamin B1 as thiamine. So here's the structure. B2 riboflavin, B3 sorry, niacin, B5 pantothenic acid, B6 pyridoxine, B7 biotin, B9 folic acid or folate, and then B12 cyanocobalamin or cobalamin. O sir, akala ko 12 B complexes yan. Bakit 8 lang? So class yung other 4, now they are not considered as B vitamins. So in order for you to efficiently memorize this, ayan, we gave a mnemonics. Tara na papi, Billy Fries, and Coke. O, diba? So feel free to use the mnemonics. So again, class, I would like to emphasize, B vitamins are not fat-soluble vitamins. All right, so now we will proceed class with our next non-hydrolyzable lipids, and those are your eicosanoids. So class, your eicosanoids, they are derived from a 20-carbon fatty acid known as your arachidonic acid. So of course, your eicosanoids are further subdivided into 4 types. We have prostaglandins, thromboxanes, prostacyclines, and leukotrienes. So prostaglandins always nag-a-appear sa board exam. So I still recall, I vividly remember na naging question to sa board exam during our year. So later on, we will discuss the specific, okay, specific info that you need to remember in our Did You Know segment. O sir, ano po yung structure ng arachidonic acid? So this is the structure of your arachidonic acid, the precursor of eicosanoids. So very varied class ang structure nila. That's why it's no use to discuss their specific structure. So remember na lang, again, that the precursor of all eicosanoids are arachidonic acid. Okay, so here is our Did You Know segment, particularly on prostaglandins. So here, aspirin works by hijacking lipid signals. So we know class kasi in our body, they utilize eicosanoids in order to send pain and inflammation signals. So that is the mechanism of action of your aspirin and ibuprofen. They block a certain enzyme that will prevent the synthesis of a specific eicosanoids, and that is your prostaglandin na nga from your arachidonic acid. That's why when we drink or when we intake aspirin and ibuprofen, nawawala yung pain and swelling. Okay? Because wala ng prostaglandins. So here is the specific reaction. No need to study this. So NSAIDs or NSAIDs, these are your non-steroidal anti-inflammatory drugs. So our example are aspirin and ibuprofen. So again class, COX-1 and COX-2 or your cyclooxygenase 1 and 2 po ang kanilang enzyme target. And that enzyme catalyzes the reaction of conversion of arachidonic acid to your prostaglandins. That's why of course, upon drinking aspirin or ibuprofen, nawawala ang pain and swelling. Again, cyclooxygenase is the target enzyme of aspirin and ibuprofen, which are both NSAIDs. All right, so now let's move forward with our third non-hydrolyzable lipids. If si eicosanoids, ang precursor molecule niya ay arachidonic acid, si terpenes naman are isoprene units ang precursor molecule. Ayan, so this is the structure of isoprene. And of course, meron tayong different types of terpenes depending on the number of carbon in its structure. We have mono until tetraterpene. Okay? Si monoterpene class, as you can see, it has 2 isoprene units because again, isang isoprene units has 5 carbon. Okay? So here are examples of your terpenes. They are readily available in nature. For instance, in lemongrass, we have citral. So actually, class, Ang terpenes, they are commonly found in our essential oils. So for 5 most common terpenes, ayan, supplementary discussion. Sorry, ayan. We have myrcene, pinene, caryophyllene, limonene from our citrus fruits, and then terpinolene. So these give the fruity scent of our fruits. All right. So now we will proceed with our last non-hydrolyzable lipid. So we have steroids. So comparing the structure of your steroids from the last two non-hydrolyzable lipids, so steroids po ay in ring structure. So it is composed of tetracyclic rings, meaning apat na different rings combined together. So here is the structure of your steroids. We have 3 6-membered rings, so labeled A, B, and C, and 1 5-membered ring. Again, class, no need to memorize the structure. And as you can see sa junctions ng ring, we have the characteristic methyl groups. So class, laging nasa 2 ring junctions yung methyl groups na yan. Alright? So here, we can see that steroids are biologically active. And actually, these are always, commonly used by our athletes. So if you're familiar with anabolic steroids. All right, sir, meron po bang precursor molecule ang steroids? So class, it is derived partly from your cholesterol. Ito class, a brief discussion on cholesterol, but we will also discuss its biosynthetic part on lipid metabolism. Okay? So again, cholesterol, as we will discuss, is a key cell membrane component. Part po siya ng cell membrane or plasma membrane natin. And of course, precursor siya ng steroid. Sir, are we capable of synthesizing cholesterol? Yes po. Okay? So, ang precursor naman ng cholesterol is squalene, which is a terpene. As you can see, guys, si terpene and steroids are somehow related to each other. Si squalene po ay 30-carbon triterpene. Okay, class? This reaction is just for enrichment. No need to memorize. Okay, as you can see, meron tayong series of reaction before we produce cholesterol in our body. Again, squalene po ang precursor ni cholesterol and cholesterol po ang precursor ni steroids. Alright, to sum up our discussion on unhydrolyzable lipids, ito class, I prepared a very short easy quiz in order to test your knowledge. Alright, first item, compounds made from isoprene. Hint on isoprene, we discussed, this is the precursor of your, yan, very good, terpenes. Okay, second item, the precursor of these compounds is cholesterol. Nako, very fresh from our last slide. That is your steroids. All right, very good class. Third one, compounds made from arachidonic acid, which then sends SOS or help signals for pain and inflammation. O, diba? Ito ang highlight ng DQK or did you know slide natin. That is your eicosanoids. And lastly, ito very easy. Ano na lang yung kulang among the 4 non-hydrolyzable? That is your fat-soluble vitamins. Again, class, consuming your fat-soluble vitamins, your ADE in excessive amounts will be toxic inside our body. Okay? And for our last topic, all right, we arrived in our last topic for lipids. So this is about lipid assembly and structure. Okay? So in water kasi or in aqueous solution, which is very typical of our cell and biological system, your lipid will assemble spontaneously into different structures or macromolecular assemblies. Sir, anong reason bakit sila may spontaneous assembly of macromolecular structure? Class, to minimize the water-lipid interaction. It's unfavorable kasi it's energetically unfavorable. Okay? That's why their tendency is to spontaneously form these 3 main assemblies: liposome, micelle, and lipid bilayer. O sir, parang same lang naman si liposome and micelle. Yes, somehow they are the same, but si liposome, mayroon po siyang aqueous cavity. And of course, they have different function inside our body. Let's just briefly discuss their functions and brief definitions. Ayan. So for micelle and vesicle, they are both spherical aggregates. Class, remember, vesicle or liposome, they are just synonymous. Okay? So again, for micelle, single layer lang siya, but si vesicle or liposome, it has an aqueous core or cavity. For functioning micelle, for solubilizing hydrophobic substances. On the other hand, si liposome, in the body or in biological systems, it takes part in endocytosis and exocytosis. So, nutrient transport inside the body. But in clinical application, it is also used in drug delivery. Okay, for clinical trials, for development of vaccines. Now, for lipid bilayer, ito very famous di ba? So this is the main component of your plasma membrane. That's why its main function is for structural support. And remember class, sa lipid bilayer, ang main component niya ay phospholipid, the most abundant lipid. Alright, remember the 3, micelle, bilayer, and liposome or vesicle. Alright, now we are on my final Lars words for lipids. So again class, here is the summary of our discussion. For the structure again, it's just made up of chains ng CH bonds or hydrocarbons. Sir, may directionality sa lipids? Wala po. It's not applicable for lipids because again, it is not polymeric. For naturally occurring forms, as we have discussed, cis isomers ang predominant in nature. But of course, trans configurations can be synthesized. Okay? Again, in nature lang po to. Energy function, yes. This gives us large amounts of energy upon metabolism. Alright? For digestion mechanism, ang enzyme niya, lipase or bile in your small intestine. So there is no digestion of lipids in our mouth. It starts in small intestine. And generally, for lipids, ang solubility nila, they are water insoluble because of their unique nature. They are hydrophobic. All right, so for classifications, we have hydrolyzable. We have three: waxes, tags or triglycerides, and phospholipids. We have sphingomyelin and phosphoglycerides. For non-hydrolyzable, we have four: fat-soluble vitamins, eicosanoids, terpenes, and steroids. They are varied in structure. And lastly, class for lipid assemblies, we have three. All right, so with that, ayan. So, this is the end of our discussion on lipids. So, for our lipids discussion, you need to answer class your practice test number 2. This is 10 items for 15 minutes. Again, similar with your PT1, this practice closed manual answering of your practice test. And then we will discuss the answers in a separate video. Okay, guys. Bye.